steps. I hope you can see me. Yeah, there we go. So thanks a lot, Rafa, Juan Antonio, Naomi, and the whole Root Benefit group for this invitation. It's a great honor to give this webinar, sorry. So I would just start with a brief introduction of the research group I'm involved in, since I'm running a small group within a bigger family, so to speak, which is what resulted from the school of Paola Bonfante here in Torino. And you have already had a webinar by Luisa and Franco some time ago. And this is the rest of the group. We are all involved in the study of basically mycorrhizals, but let's say on a broader scale, beneficial plant interactions with different shades. Some of us, like Elena or Sylvia, are more devoted to ericoid or orchid mycorrhizas, whereas the bigger group, I would say, is on arbuscular mycorrhizas, and some of us also study ectos. But basically, we are all into the plant symbiosis with different shades again. So some more basic research, some more applicative research, also depending on where the funding comes from. We have to orient our interests in that. But yes, you can find more information at the link here below, which is our department link, and you will find all of us there. So let me just step into my my topic today. As we all know very well, plants interact, and in particularly the roots, or the root system of plants interacts with a high number of different microbes. Many of them are bacteria, but fungi also play a major role in this microbiota, this root-associated microbiota. And coming to symbiotic fungi, which are our topic of interest today, they are attracted by root-exudated strigolactones in particular. The perception of these molecules triggers several changes in hyphal development, including a higher number, more intense branching, so the development of a higher number of hyphal tips, which is a basic, probably, strategy to improve the chances that eventually these tips will get in contact with the root surface. And this is what is happening at this stage here with the development of a hyphopodium, which is a large hypha attached to the surface of the epidermal cell, which is a first step for entering into real symbiotic interaction with the colonization of the inner tissues. In fact, around this stage, the mycorrhizal fungus is also releasing its own signals. We call them mycorrhizal factors, and they activate symbiotic responses in the host cells, which in due time lead to successful colonization of the internal tissues, the development of arbuscules that we see here in the cortical layers of the root, which are the main site for nutrient exchanges between the plant and the fungus, with the fungi providing mineral nutrients to the plant, namely phosphate, nitrogen, and receiving in change organic compounds, especially sugars and lipids, on which they are completely dependent on the plant supply. The situation is quite different when a different type of fungus interacts with the root. Pathogenic fungi trigger defense responses, but then the outcome is completely different, as we know very well. So in spite of this striking difference between the two responses, it may be surprising to see that a key role in the perception of both types of interactions, mycorrhizal fungi and pathogenic fungi, is based on derivatives of the same macromolecule, which is chitin. It's a polysaccharide that is composing, it's a major structural component of the fungal cell wall. And what was initially hypothesized, and still holds true with several studies in the years, is that short chain chito-oligosaccharides, so short derivatives of chitin, versus long chain COs, characterize symbiotic versus pathogenic fungi. Indeed, several studies have identified plant receptors that are able to recognize short chain COs versus long chain COs. And I said receptors, but I should say receptorial complexes, because as you can see, several proteins collaborate for the perception of these short chain or long chain COs, with some elements that can be shared between the two types of complexes. Okay, so far so good, except that it has been very clear since the beginning that both mycorrhizal fungi and pathogenic fungi actually produce a mix of COs of different length, both long and short chain COs. So this is complicating things a little bit. But in the same period, other groups also highlighted that mycorrhizal fungi produce a variation of these chitin-derived molecules, lipocyto-oligosaccharides, where the chitin backbone is added with a lipid tail. And this specific molecule, which resembles very much the rhizobial knot factor, is perceived in mycorrhizal plants by specific receptor complexes, which include some elements, again, that are shared with the MIC factor perception. But this would give us, how can I put it, a reinforcement of a symbiotic signaling specific for mycorrhizal fungi that would not be present in pathogenic fungi. Again, so far so good, until someone discovered that LCOs are actually produced by all analyzed fungi, including pathogens. So what is really going on here? It's clearly kind of puzzling, this situation. But let's try and look into that with more detail. One thing we should consider is that for the COs to reach their receptors on the plant surface, on the plant membrane, they must dissolve in water solution. And here we see a major difference between short chain COs and long chain COs in terms of solubility. Short chain COs are easily soluble in water, whereas long chain COs are quite difficult. In fact, when you want to chemically analyze the two types of molecules, you really have to use different solvents, because hydrophilic, well, polar solvents will not work for long chain COs, even just for CO8. This means that probably short chain COs can more easily and possibly more quickly reach the receptors, whereas long chain COs will arrive with some delay, with some more time, or more difficulty. And indeed, in this line, it was shown that pathogens maybe have an interest, if we can put it this way, to humanizing them a bit, in not alerting the plant too soon that they are coming. Indeed, several pathogens have been shown to produce specific LISM molecules that capture their own long chain COs, preventing their association with plant receptors, so somehow masking the presence of these long chain COs. And indeed, in a similar scenario, it was shown that AM host plants, this was Medicago if I'm not wrong, produce an analogous molecule, protein, that binds long chain COs, and this molecule is required for correct arbuscular development. So again, quenching defense responses in this case can promote symbiosis. So this aspect is also very relevant, most likely. Okay, let's take a step beyond the surface receptors in the plant membrane. What happens then? What happens next? Well, it was long acknowledged that repeated oscillations in nuclear calcium concentration, what we call nuclear calcium spiking, are typically associated with symbiotic responses. In response to both COs and LCOs, all epidermal cells start flashing in their nuclei with these peaks of calcium concentration. By contrast, the perception of long chain COs is related to diffuse, a broad and singular and individual transient elevation in calcium concentration in the cytoplasm, which you see here in the bottom of my image, and that's associated with MAP kinase activation, reactive oxygen species burst, and the regulation of pathogenesis-related genes, and in one word, immunity. So again, we had a kind of a dogmatic view of this until new research showed that long-chain COs, when applied individually, can also trigger nuclear calcium spiking, and short-chain COs can also trigger a cytoplasmic increase, transient increase in calcium concentration, which could be related to the ability of mycorrhizal interactions to prime plant immunity, which is involved in the fact that the mycorrhizal plants are less prone to pathogenic attacks. But again, we are starting from a nice and clear division and ending up with a mixed picture, which I try to schematize here, where we have at the top the different types of molecules that vary in length, first of all, which is a major difference, which is related to their solubility in water. We also know not so much about their actual concentration when they are released by the fungi and perceived by the plants, especially their relative abundance. As I mentioned, it's difficult to extract and quantify long and short chain COs in one run, so it's not always easy to understand how much of each species of molecules is present in fungal exudates when plants are receiving them, are perceiving them. Another important aspect is the acetylation degree. We have seen that these molecules can be more or less acetylated on their monomers, and the more they are acetylated, the more active, biologically active they are, at least as symbiotic signals. And then, of course, there are substitutions. So this lipid tail or other substitutions that are present in LCOs, for example, clearly add a level of complexity here. Then we have the receptors, where, as you have seen, some individual proteins may play a role in both receptorial complexes, both related to pathogenesis or symbiosis. So there's also probably a competition between which molecules will activate an individual receptorial complex and not. So you will have a receptor in one complex, and that receptor will not be able to form a complex in the other pathway. So this is, again, difficult to grasp. Already mentioned about the calcium signal issue here, where we have cytosolic and nuclear calcium signals. And we should not forget that this is one or two parallel signaling pathways that we are talking about, but additional pathways may be running parallel to this one, like additional elicitors released by mycorrhizal fungi or pathogenic fungi, that is well known in pathogenic fungi, that may be activating other responses that altogether lead to symbiosis versus defense responses. And another interesting hypothesis that was raised a few years ago by Giles Oldroyd was that the plant nutritional status may also play a major role, with well-fed plants being less prone to root colonization, no matter what fungus is coming, and starved plants opening their cells to anyone basically in search for help from mycorrhizal fungi, but then allowing also pathogenic fungi to attack them, which is, again, interesting and will have to be demonstrated and investigated further. So I think you are convinced with me that this is a big, complex situation. Most likely a complex solution that plants and fungi have found to manage a complex problem. And what I want to stress is that this is very complicated for us to understand, but it's apparently not complicated at all for the plants, because as soon as you treat a plant with short chain COs, as you can see here on the left, you have a higher degree of colonization by arbuscular mycorrhizal fungi in the root. And if you treat the roots with long chain COs, you have a quenching, a weakening of the symbiosis. So the effect is very clear cut. It works. Right? We are not able to really understand everything, but plants do. So let's keep in mind that. So based on this knowledge, we have now been starting since a few years to develop some applicative studies, and I wanted to present you with some of the latest results we have here. We initially had a study on alfalfa, where we were treating these plants in an open field with the COs and checking their mycorrhizal status and their production. But now we move to a major crop, tomato. So basically, we have been treating the plants with short-chain chito-oligosaccharides and a commercial inoculum. And then we wanted to check the symbiosis, the effect of the CO treatment on symbiosis development, on plant development, on its resistance to a fungal pathogen, and the quality of the fruit. So to give you a view or an overview of our experimental setup, we basically have 4 conditions: control plants that have not been treated, CO plants, which are those that have only been treated with chito-oligosaccharides, M plants, so that is mycorrhizal inoculated plants, and MCO plants, which are mycorrhizal plants that have also been treated with COs. And this has been done in the field and in the lab in parallel, distinct experiments. What we have seen is that, as expected, CO treatment in mycorrhizal inoculated plants led to an increase in the colonization of the root system by mycorrhizal fungi. Several parameters are significantly increased here. And also in the field, we have some increase in a trend of increase in fungal colonization upon CO treatment. And this is particularly evident here, but what I wanted to show you here, to spend just a minute on that, is that, of course, when you work in the field, you also have a the native presence of mycorrhizal fungi. These fields, unfortunately, have been treated for decades with fungicides, so the population of mycorrhizal fungi there is probably quite weak and poor. And indeed, we do not see an effect of the CO treatment in the control lines, where there has been no inoculation, so just the basic native presence of mycorrhizal fungi in the soil. Whereas the inoculated fungi seems to respond more actively to the CO treatment. So the effects on plant performance, we have seen an increase in plant height, in plant biomass. The difference is always very significant between MCO and control plants, but also mycorrhizal plants already have a positive improvement in height, biomass, and also the number of fruits. So we have an improved plant performance associated both in the field and in the lab with the mycorrhization and CO treatment. And then, as we were sampling our plants in the field, we realized that since we had asked the farmers not to use fungicides, at least for the period of the experimentation, inevitably some pathogens showed up, and indeed we found an alternaria infection in our plants. But we remarked that the number of infected plants was different. So we simply counted how many plants were infected per row in our experimental fields. And it was striking to see that control plants were much more infected than CO-treated plants and then mycorrhizal and MCO plants. So we did see a very macroscopic effect of the treatment on pathogen infection. So we wanted to look into this with a little bit more detail. So we repeated the experiment in the lab. So now we have, again, our 4 conditions, but duplicated with non-alternaria infected and alternaria infected plants. And what you see here in terms of root colonization by mycorrhizal fungi is that the CO effect, so the effect of CO treatment on the intensity of mycorrhizal colonization, is particularly important in infected plants, the brown ones here. Suggesting that the suffering plant is somehow trying to rely on the symbiont for help. That's a suggestive view, if you want. What we also did with more detail in the lab was to measure what pathologists call the disease index, which is an estimate of the extension of the infection in the leaf. And we can see that both mycorrhization and CO-treated mycorrhization in infected plants drastically reduced this disease index, which is basically the size of the lesions on the leaf. So the next question was then, why are these plants less affected by the pathogen in the presence of an intense or powered up mycorrhizal colonization when we use the COs as well? our first hypothesis was that maybe the leaf cuticle was, or the cell wall as the first line of defense for plant cells, was stronger in these plants. So we did a very simple experiment of sectioning tomato leaves and then staining the root and cuticle with different dyes, fluorescent dyes. And you can see here the results. We could really measure the thickness of the cell wall and the thickness of the cuticle. And what we expected was that, just in a very naive way, maybe the cell wall was thicker or the cuticle was thicker. Surprisingly, the results was the opposite, that CO-treated mycorrhizal and MCO plants all had a significantly thinner cuticle compared to control plants. This was very solid and surprising, but a result. So, of course, cuticle thickness is just a very macroscopic information about what is happening in the cuticle. So we thought we could see something more, something clearer, maybe by looking at the regulation of genes involved in the synthesis of cuticle or waxes. And again, we have a significant change in gene regulation pattern for these metabolic pathways, but we have upregulations, downregulations. So the picture is not at all clear, but what is very clear is that there is an important effect of the CO treatment and of the mycorrhization treatment on the composition possibly of chitin. This is what we are missing, of course. We could not do a biochemical analysis of cutin for the moment, but there's a change in thickness, there's a change in gene regulation. Coming now to fruit quality, we wanted to check whether mycorrhization and CO treatment had an impact on the quality of the fruits, because these are tomato, what they call industrial tomato. They are produced to make tomato sauce, basically. So we use this rainbow protocol, which is a colorimetric assay, rather, I wouldn't say easy, but faster than a detailed biochemical analysis, to give you indications on some groups of metabolites. And for example, we could see that the number of the amount, the concentration of free total amino acids is increased in both mycorrhizal and MCO plants compared to the controls, which impacts on the nutritional value of tomato. In particular, proline is used as a proxy of plant health or absence of stress. And indeed, again, mycorrhizal and MCO plants have more proline in their fruits than control plants. We also checked the Brix degree, which is the amount of sugars contained in the tomato sauce, which is really the requirement for selling your tomato to the producers, to the work that produced the sauce, I mean. And in this case, the difference was not so striking. There are significant differences, but all of the four conditions are well above the 4.5 threshold, which is what is required by law for using tomato for this production. So that was okay. The last parameter we looked was the content in starch, which is again interesting because starch is not normally what you want to have in your tomato, and it's actually being accumulated in the fruit before maturation and then being cut down into simpler sugars as maturation proceeds. So what is interesting here is that MCO plants have a smaller amount of starch in their fruits, suggesting that they are in a more advanced maturation stage compared to the controls. So to conclude, we have treated our tomato plants with COs. We have seen a positive effect on symbiosis. We have seen a positive effect on plant growth and performance, productivity. We are investigating fruit quality now with more detailed biochemical investigations, but the indications are interesting. We have a reduced infection by this pathogen, and we have seen changes in the cell wall structure, even if these are more challenging to really understand completely. Okay, with this, I think I have almost concluded. I just wanted to thank you very much for your attention and all the people involved in different aspects of this research. And I will just mention that the video I showed you at the beginning is available at this link for download. So please, I'll be happy to take your questions. I didn't check the time, but I hope I'm more or less on time. Okay.
Yeah, thank you, Andrea, for the very, very nice presentation and also for including so many new and exciting results as well. So if anyone has a question, feel free to either type them in the chat, or if you join as a speaker, activate your microphone and ask them. I can see that Juan Antonio also very excitingly already asked a question about the fertilization used for the tomato field experiment, since we know that nutrition is, of course, very important in symbiosis establishment. Can you say anything about...
Okay, so yes, we asked the farmers not to change anything in their normal practices in the field, because we wanted to check whether AM inoculation and CO treatment had an effect under normal agricultural conditions. So the only thing we asked was not to use pesticides, not to use fungicides specifically. I think they did use pesticides for insects, aphids, and so on. So the fertilization, we have some soil analysis giving us the detailed information on how much phosphate, nitrogen, potassium are there, but it's just regular fertilization that was used by the farmers before we came, if that addresses your question.
It's already very nice. I can also see someone asked if the presentation file will be available. We do publish everything also on our YouTube channel, so you can also access the link there. I can also see
this is all unpublished results, but I'm not afraid of publishing.
Okay, yeah, but we can always discuss.
Please take care.
Yeah, will do. Then there's also Stephanie, who has a question. She might have missed it, but what was a pathogen you used as a biotroph or a necrotroph? Yeah.
It's a necrotroph, actually. Yes, I think it's a necrotroph. Alternaria alternata, yeah.
Okay. Yeah, feel free to ask any more questions if there are. I also had a question about the beautiful image that you had as well with the cuticle modifications that you used to quantify the sizes. How did you stain them specifically?
That was stained with calcofluor for the cell wall and with Auramine O. Auramine O, which is a specific stain for a specific dye for WOXs, basically lipids, and so you can have this nice two-color staining in the cell wall. It works smoothly. Yeah.
Very beautiful. Yeah.
The leaf explants were fixed and then sectioned in a vibratome to get these transverse sections.
Okay.
Here you have the blue. That's the autofluorescence of chlorophyll, of course.
Yeah, it's a very beautiful image.
I also have a question, if you don't mind. How do you treat your plants in the field? I saw the spray, in the jar.
Yes, that was not explained. You are right. So we use we spray the solution in the lab because it's easier. In a small pot, we just spray the CO solution on top of on the surface of the soil. But for field treatments, we normally, based on previous experience, we prefer to do the treatment at the very beginning. So when plants were produced in the how do you call this? Well, in the greenhouse, basically. Yeah, in the nursery, they produce these tiny little plants of tomato, and as they were sown in these little volumes of soil, that soil was already treated with or without mycorrhizal inoculum and COs. So they were treated in a clean, small environment, if you want. And then there was no additional treatment, so they were just planted in the field, and that was it. So what was done was all at the beginning. This is not surprising because in a previous publication we already saw that, in the previous study, we already saw that the effect of the treatment is visible like months after the application in the field. So probably bursting mycorrhizal colonization in the beginning is sufficient for the plant to maintain that afterwards. So yeah, that may be surprising, but it's
yeah. You think it will be, let's say, you will have an additional effect if you treat it later in the soil or in the field?
It's possible. We haven't tried to do that yet because it is not so easy. Once you have the plants growing, adding this treatment will go on the leaves, and then COs on the leaves are known to induce a range of effects, also involving pathogen defense, etc. So we might be confusing things a lot. So for the moment, we haven't tried that. In the previous study, we were working on a grass field. So they were growing grass for hay, basically for animals. So periodically they would cut the grass, and in that case we did add occasionally some CO and inoculum treatments, like at the end of the season for the next year and so on. So we could repeat that when there was no leaves around, basically when the grass was cut. But for tomato that was not possible, of course.
Okay, thank you.
Thank you.
I have a small question. Great talk. Thank you, Andrea.
Thank you, Olga.
Do you have an explanation, and I don't know if you know at all, about why the addition of COs didn't increase the colonization by native fungi, but increased the colonization by the inoculated AMF?
Well, this is a bit surprising. We didn't see that in the grass field, for example. But a grass field is a much more natural, nature-like environment compared to a field of tomato. I don't know if you've seen what a tomato field looks like, but it's completely bare, and there's just the tomato plants. So my interpretation, is just a speculation, is that the few survivors of mycorrhizal fungi that are present in that soil are probably either too few or too weak after, again, decades of fungicide treatments to efficiently colonize the roots. We have a low level of colonization. Where was that? Sorry. Yeah, here, 25% is not much. We normally have at least 50 or more. So probably that's the best they can do. And the CO treatment, which is stimulating the plant, not the fungus, is not probably doing enough for the native population. Again, this was a bit surprising, but because we did not see that in the grass field, where the native population was more active and was already giving a better colonization with the CO treatment alone. Nevertheless, I would expect this to be caused by the weakening of this mycoflora by the fungicide treatment. That is my interpretation.
Thank you.
Where again, the other case, the mycorrhizal plants were treated in the greenhouse, so in a way, in a very protected volume of soil, and then moved here later on. So this is a major difference, probably.
Okay, I see in the chat at least there's no more questions. If there are any more questions, feel free to pose them in the meantime as well. But I think it would be timely also to move on to our next speaker as well.
Absolutely. Thank you very much.
Thank you very much for a very nice presentation.